Thank you. I think we've got 48 individual computers online. I'm hoping, and I think there probably will be many more than 48 wanting to hear your talk. So everybody, it's my pleasure to introduce Shawn Kefauver, who's a research fellow in agriculture and forestry and a professor of plant physiology in the Faculty of Biology in the University of Barcelona in Spain. Shawn's got a number of research interests, which range from ecophysiology to phenotyping using various types of image data capture. And I understand that he has a particular interest in precision agroforestry and scalable high throughput plant phenotyping. I'm not going to say much more, except that today Shawn's going to present a summary of his research over the past few years on phenotyping cereals using a range of image sensors, and explain a little bit to us, some of us who don't know very much about the role of AI and machine learning in estimating things like yield and quality. So the title of Shawn's presentation is Towards AI and Cereal Phenotyping, and I'll pass over directly to Shawn. Welcome. Thank you for joining us.
Thank you. Thanks for the invitation and the great introduction there. So the title Towards AI and Cereal Phenotyping, since it's the International Barley Hub, and I don't do a whole lot of barley research, but we've dabbled a little bit sometimes here and there. So it's about cereals, but a lot of the stuff is applicable or useful towards barley. And well, we've just recently kind of been doing getting to the point where we can do machine learning and AI, and it's been a bit of a journey. So I thought I would share that journey with you. I'm a researcher at the University of Barcelona, also here signing as Agrotechnio, which is our sort of joint university research foundation, which is pretty popular these days in the Catalonia region of Spain, which is not where I'm from, but definitely where I've been working for the last 10 years or so. Quick. Well, that's about it for myself. Let's go into artificial intelligence. I grabbed this from the web. If somebody made a nice graphic sort of outlining things here. So a lot of people talk about artificial intelligence is actually an old idea, but I think is something that's become more and more practical through machine learning and through deep learning. So technically, deep learning would be a subset of algorithms based within machine learning, which is within this overarching concept of AI, artificial intelligence. And deep learning sort of being this multi-layer neural network subset that has a couple of things that are quite interesting. What is artificial intelligence? So a couple of categories about different types of models. We have supervised learning. So I would say that artificial intelligence is usually supervised. You usually wanted something specific. There is some aspect of artificial intelligence that could be called unsupervised, in which you let things sort of freely learn. So this is somehow these giant AI machines that are supposed to develop free thought after some time. So they just gather information, but they're not looking for something specific. This could also be, these ISO data, these older sort of models where unsupervised learning, like classification or something. And then this idea of reinforcement learning, I think is something that's been expanded on within the concept of deep learning, which is quite interesting, which is optimizing the solution. So you gather lots of data, you analyze it, reanalyze it, and you try to optimize the solution as much as possible. Just as an example of a reinforced learning, for example, in optimizing a solution, I once used, I'm not going to present it here, I once developed a machine learning algorithm to count pine cones. It was just this little side project in forestry. And the precision was really high, but it would take like 12 hours to count a dozen pine cones in a tree from a mobile phone picture. And we made a fun little mobile app to count the pine cones, but it wasn't very optimized. You could count to 12 before the algorithm would finish doing all of its models and stuff like that. And I think the reinforcement of deep learning is what's really gotten us to do that. So when we ran the same theme through like a yellow V5 deep learning. It did the same. So again, these are also old concepts to new concepts. So people got excited about robots, even going back to the 50s, and of course, things didn't really move very quickly until we started collecting more and more data. So this idea of big data kind of came about, and I think in a lot of ways, especially if anybody's working with drones or capturing data with mobile phones, sometimes you end up with too much data, you don't know what to do with it. So sort of the machine learning and the deep learning are these mathematical concepts, and here I'd like to throw out a little caveat. I'm not actually, wouldn't call myself an expert in machine learning and deep learning because I'm not developing new algorithms. I'm developing new uses for existing algorithms that have been thought up of by more mathematical minds. So anyways, so AI to be effective and efficient needs big data. So then we get to the point of how can we make big data? I mean, unless you're just trolling for pictures on the internet and gathering a million pictures of cats and dogs for your algorithm, you'll need some collaborators and some time. So this is a bit of my story through collaborations and time to try to get to that point. And the field of application in which we did this was phenotyping. Here, phenotyping, we outlined in one of our review papers a couple of years back, sort of combining different platforms, different sensors. So it might be a digital picture or some thermal or some spectral information, different platforms, whether it's you on the ground or drones in the sky, how to process and curate the data. Of course, the environmental characterization and spatial variability is important for phenotyping, but integration has been a big bottleneck for us. And so we've been looking at different ways of integrating and processing all this information and phenotyping. I think phenotyping itself has always been sort of a numbers game. But then with the integration of drones and more new technologies, the microplots that we have in phenotyping have sort of exploded with the amount of information we can extract from them. So here's some examples of low-cost sensors and affordable platforms. Here presented somehow in Technicolor because I don't know, my screen, this looks really bright. But yeah, we try to catch the eye sometimes with these things. So thermal sensors can be a thermal gun. We had a mobile phone capturing thermal data. We have drones capturing thermal data and spectral data. We have field sensors capturing multispectral data. I've found and became a big fan of red, green, blue RGB images, just digital photography for quantifying color and spatial aspects. And then different platforms. Here you see, of course, the drone option, putting your camera on something that flies is definitely a lot more appealing than the Phenopole, which you can see me holding down below. Of course, you can't fly drones everywhere. So sometimes we use the same cameras on these big telescoping poles, and we just sort of run through the fields, making little whirring noises to simulate what would happen if we could fly. So this is the information that we're gathering. Again, the more low cost it is, the more affordable, and the quicker to process, the more data you can get. And we get closer and closer to that big data. Quick comment about RGB images, which I've been become a big fan of. We've seen that we can get this data from pictures on the ground, from mobile apps. We found that it's quite well calibrated for color, and that in some instances you can substitute some of the extra spatial information from RGB can replace some of the extra spectral information that you might get from 3D cameras. Which you can see here in the, see if I can get the laser pointer function here. Boom. And for example, there's some small differences between aerial and ground, but you can get very high resolution cameras. At very high resolution, we can also segment things. We can enhance contrast and segment even plants at early stages. Of course, if you're flying from a drone and you have that aerial perspective, you can apply these structure from motion models and create 3D models of your crops. This is just a funny example where I tilted the camera at 45 degrees and flew around in the perimeter of a study plot, and it aligned all the pictures. So these models are quite powerful, and they do a lot of the processing for you. In terms of disease monitoring, up on the right-hand side, we've seen good precision with quantifying changes in pigments. Again, this is color quantification, which we can do at high resolution. We can track chlorosis and necrosis related to diseases. And then also segmentation capacity. I'll talk a little bit about spike density kernels. And here we have some applications in barley with ear counting as well. So here's some pictures of the scales. I mentioned platforms. So when we say platforms, that also goes to scale. Each platform captures data at a different scale. So we've been exploring leaf scale, ground scale, and aerial scale. You can see this is actually a wheat platform, but you can see instantly big differences just from an aerial picture here being rain-fed and with supplemental irrigation, a giant change in color. So RGB, so if you look at the full near-infrared spectrum, here you have red, green, and blue, or blue, green, and red, and you see differences between soil, unhealthy vegetation, and healthy vegetation. Of course, some of these differences seem like they're bigger in the near-infrared, but the truth be told, that there's still differences in red, green, and blue, and these are more affordable, more available, faster processing, higher spatial resolution cameras for the most part. So all of those other aspects of these cameras can help us offset what we might be missing in the near infrared, especially if we think about how to process it. And then if you're interested in pigments and photosynthetic efficiency, look like I got cut off a little bit here. I'll have to adjust that. So photosynthesis rate, and here this is absorption, and from 400 to 700 we have blue, then green, then red, and you can see exactly where our photosynthetic pigments are absorbing light here. So again, if we have good color, good coverage of PAR and high resolution, then we can collect data that's going to be relevant, and these cameras are quick. So here's a couple examples of our RGB journey. So this is the one I mentioned beforehand, looking at lethal necrosis in maize. Related to this, we developed an index assessment of maize lethal necrosis, and we found, again, all of these are from hue to green or green area. Everything except for the red one. These are all different indexes based on RGB cameras is here compared to sort of NDVI as a scientific standard. I think somewhere I have, in case I forget about it, we developed a piece of software to calculate these RGB vegetation indexes for maize, and we called it the maize scanner, basically because I have very limited powers of imagination in some cases. That's available. I think there's more about it later. We tested it with yellow rust and wheat. Again, this seemed like a pretty obvious opportunity. So you can see here high resolution color image, and we see a surface change in color here. So we thought we could quantify that, looking at different growth stages, and we can see that again the RGB is as good like here in heading, or even much better in determining grain yield in wheat for yellow rust impacts. We also looked at nitrogen stress in maize. This is another study here, nitrogen prediction. This is leaf chlorophyll content, I believe, calibrated with SPAD, leaf nitrogen percentage, and then if we look at the performance of here, the greener green areas and RGB index, again, we have a pretty good correlation when we're quantifying the amount of green area or the greenness of the vegetation area that's here. So these were comparable to a leaf sensor of chlorophyll, the RGB indexes, and outperformed NDVI. Again, NDVI for nitrogen stress might confuse biomass with chlorophyll, since it's kind of a combination of both. So moving forward and thinking about how to collect more data and produce better results, we had a collaboration with a company here, and we started putting together these tools into something that would be easier to use and more convenient. So here we developed the Cereal scanner as part of a PhD with Jose Fernandez Gallego here, a couple of his PhD chapters, developing specific tools for early vigor, ear counting, which I think we also tested on barley, and it worked quite well with a little adjustment. Maturity assessments, and then some of these different vegetation indexes that we liked at the time. So this was 4 or 5 years ago. We used this breed picks combination of indexes, including the green area, greener green area, and senescence index, which were developed by former researchers in the lab, and a bit popularized by myself, and then also in some collaborations with our colleagues at CIMMYT in Zimbabwe. And I added on the normalized green-red difference index and the triangular greenness index that were developed by the USDA in the United States, because I really liked the concept that they applied, and these 2 papers here, which I cited. We did a video journal of visualized experiments, video tutorial use on this software, and it's also open source, so check it out. The Jove was kind of fun to do, explaining how to use it using field cameras. So that's there for you if anybody wants it, if anybody wants access. It should be open access. If you need help installing it, just let me know. So here's the Cereal scanner. Abiotic stress conditions tested for wheat ear counting. The system is based on a combination of different filters, Laplacian and medium filters, and then it does ears per square meter, which of course you need some camera specifications, focal length, distance between the canopy and the camera. And then it gives you not just the ears in the picture, but the total spike count. One thing we found that it's a little bit dependent on this technique on growth stage. So here performing best when the spikes are a little green and the leaves are a little yellow, and there's a little bit more contrast. This was a study of early ground coverage that we did here, how to optimize these algorithms to best segment out the amount of ground coverage of a crop in early growth stages when the crop is quite small compared to the ground background. Here we developed the image database using Phenopol, so camera on a stick, and here the best results sort of came from the CIE Love Color Space, which is from the International I think the acronym is actually in French or something like that, but it's a camera photography calibration used by professional camera photographers, digital camera photographers, and it's really good at separating out these axes of color from the intensity of light in the image, from white and black. And so that was really great for segmenting green leaves, whether they're in the shade or in the sun, because it separates light from color. So after the Phenopolls, we got drones, and we started collecting more and more drone data, first RGB pictures, then later adding on multispectral and thermal. And then we found ourselves spending months and months post-processing the data. And it was great to collect the data too quick. And then it took us months and months to process it afterwards. So we went from collecting data to pre-processing it to collecting data again first year. The Mosaic tool kind of helps automatically segment the plots, or semi-automatically segment the plots. with a little bit of time for adjustment in between. And that brought our processing down to about 6 weeks for cereal crops. So in a nutshell, you kind of put in the plot size, dimensions, the number of replicates, and the numbering pattern, and then it gives you a pre-drawing of your field layout with all plots the exact same size. Then it gives you an option to adjust them a little bit and export the microplots for processing. This is a plugin which functions within Fiji, which is also called ImageJ, which I think was originally developed by the National Institute of Health of the United States. And one of the reasons we originally selected it was, one, it looked like it was good for diseases. Two, it was open source and it had a good following. So we thought it would be continuously developed and improve over time. One caveat, I think Fiji has now outpassed us, and the newest version of Fiji might not be compatible with all of our plugins. So we provide an old version of Fiji that now is compatible with our plugins, while we're trying to figure out how to update the changes that they've made over time. Just to throw that out there. So then we started comparing aerial scale things to see if there were big differences. Here you see this is the R-squared comparison of water stress in wheat, yield prediction. Here we saw a slightly better performance of NDVI in the aerial scale, but just a little bit more than RGB indexes, whereas the ground scale RGB seemed to perform a little bit better. So this is sort of an indication, our first indication, that maybe the spatial resolution was limited. If we use the exact same camera on the ground as we did on the drone in the first days, so we tried to use that camera only on the ground scale and improve our aerial cameras so that the resolution is not a limiting factor. This was sort of the beginning of multispectral studies. I threw up the International Barley Hub logo on this one because this was actually a barley study. This was a collaboration with a company where they wanted us to test these phenotyping platforms on this lovely little plot here at the base of a castle in Spain, which we quite enjoyed. You can see here the green area and the greener green area. I don't know if those are fuzzy for you. I'll have to check the quality of that graph there. Performed quite well in estimating grain yield. The crop senescence index later in the season also performed quite well. And more or less, I would say the same or slightly better compared to some multispectral indices, these being SAVI, the Soil Adjusted Vegetation Index, which combines near-infrared, blue, and red, I believe, and OSAVI, which was a slight modification, the Optimized Soil Adjusted Vegetation Index. So at this point in time, we were flying also multispectral cameras on our drones, and so we started comparing these. Now you got to keep in mind that this is a bit multispectral on the right compared to RGB here. But we probably had 20 megapixel.
Also might have a task of very I will decide because at the point you will have the fiber and the solid, they will separate. But if this process with what I mentioned at the beginning of, let's say, integration between the two during the composition, means that the better will be.
Interesting. Was that a question for me?
No, that was somebody who was unmuted. Sorry about that. Sorry.
Oh, okay. I was trying to follow along and I didn't catch all of it. Anyway, so the multispectral index is over here. We're done at 3 megapixels, right? So there is it's not really a direct comparison because the spatial resolution is so different between these. And we took it to Africa and we continued testing ground and aerial and pushing the limits on what this could do. So this was low and optimum phosphorus treatment comparison here in, I think this was in Zimbabwe. Even at early growth stages, we found quite good comparisons here. And even when the multispectral images had lower resolution, they still performed similar to RGB. Of course, considering that the multispectral camera costs 25 times or 50 times more than the RGB camera, you have to take that into consideration. This was another one. So RGB only comparisons. Here we did see pretty good function with RGB, and then we found, better compared to NDVI. A little bit less than SPAD. SPAD again here being a leaf sensor was quite different, and it really captures chlorophyll better because it's absorption, not reflectance. And we had good results, especially when combining that with some other field metrics such as flowering time that were important for maize. So the last example here was our project Wheat Phenoscale. We had this great idea to take it up to another platform, to another level. So what we did is we took our 11-band UAV multispectral camera here at Tetracam Micro MCA, and modified the bands so it would be a simulator of the Sentinel-2 satellite right when it was launched. We started collecting data in phenotyping trials to develop machine learning models that we could then apply to satellite data from Sentinel-2 at 10 and 20 meters per pixel across large fields. So trying to use, leverage the microplots of phenotyping to apply in a precision agriculture concept. So that was the idea of Wheat Phenoscale. Here's the two papers here that came out just recently on that. And this would be sort of my concept here. This is a kind of a blend here of combining the results from the two papers and one sort of project process concept. So we looked at the workflow of UAV microplot segmentation, dividing that up in both sets, and training and testing data, fine-tuning neural network, deep learning or machine learning algorithms, looking at which perspective, which levels produced the best correlations or the highest positive or negative correlations with not only yield, but also nitrogen content and some other properties, and then applying those to actual pixels of grain yield, estimated grain yield, and grain nitrogen content that were collected by a combine harvester with an integrated GPS, and it was producing yield and quality metrics on real time. And then testing that not only against grain yield, but also looking at protein content. And here we even tried to get a handle on vitreousness and test weight as well. So that was the Wheat Phenoscale project. That was kind of this ramp up of how can we best improve processing, image gathering, optimization, and get enough of these microplots to be able to feed into an algorithm that will really train a model and be able to scale that up to a different satellite by developing a UAV simulator. So so that was the sort of the culmination of the Wheat project, and we've been doing some other things, trying to get our hands along big data and how to process big data in international collaborations. So we started playing around with the Open Data Kit. We developed a couple pieces of software to try to leverage Open Data Kit, which is a mobile app that allows you to capture images in the field that will put together all of the data into an aggregate server. And then the ODK downloader allows you to download it or reorganize it and feed it into a deep learning algorithm. So ODK, if you don't know, check out the website. It's kind of a fun, it's a little mobile app with a form you can build up. It's used all sorts of people all over the world. And it's great for offline data collection, which is great for collaborations in developing countries, because they can take all the pictures in the field, and then when they want to upload it, they pop into a cafe and they can upload their forms. You can add pictures, which of course, as a remote sensing specialist, that's what I'm interested in the most, but you can also add GPS locations, calculations, and multiple language support. So we threw out a couple of ideas how to use ODK aggregate. So you can collect the data. They help you develop these servers to put together the data. We can process the data in the cloud and produce RGB vegetation indexes, leaf area index assessments with fisheye attachments. We can feed that back into a fusion table. I think it's now called Google Sheets, and get real-time mapping functionality, or we can also feed that into Google Earth Engine analyses, looking at the verification and the incidence of disease assessments or something like that once it's been spread. Here, this is, I think, fall armyworm across Zimbabwe. We developed a couple of PC-based tools using Java, which will automatically download the data, process the images, and give you the results of the image analysis and allow you to visualize it here. The Fusion, which does the prefix algorithm, and here Cereal Fusion, which actually gives you the option to process all of the versions from the Cereal scanner integrated into the ODK environment. We tested it across 3 countries in Africa for monitoring the fall armyworm, with pretty good results, at least just enough, I think, to be able to make a nice publication on this collaboration with the FAO, and they took some of our ideas and they've been using some of them and implementing in their own global platform called Famous. I think this could also be useful, of course, for barley, otherwise I wouldn't be sharing it with you today. If you have disease incident specialists or geographers who are interested in these sorts of things, you could piece it together. And then lastly, we had this big collaboration trying to map different diseases. Obviously not on barley. I think this is a picture of a cucumber with some infestation in it. So we developed this ODK downloader once we started collecting thousands and thousands of pictures. It's just a way to reorganize it into a different directory, file name, and an image structure. So these are tools that I think would make available to anybody who thinks they may be useful. So that's it for my talk. I hope I was within time there. If you have any questions, here's my email, the group website, my personal website. Here we have a list of collaborators. Some of these are former PhD students who have moved on to doing wonderful things elsewhere. And just to finish up, I also have cost action, like Sonia, who wasn't able to make it today, where we are focusing on remote sensing applications in phenotyping, precision and regenerative agriculture, and sustainable land management. This is the nexus of these applications and these technologies. We're finishing up our first year now, which means we have 3 more years to go. You are welcome to check out our dynamic website here, pangeos.eu, or our official website with the sign-up links at cost.eu action CA 22136. And of course, also there, if you want to reach out to me, I'm the chair. My information is listed as well.
Shawn, thank you.
With that, I think close my screen unless somebody wants to see. Wait, there we go.
No, Shawn, thank you very much. That was really informative and instructive. So there's probably around 70 people online, and if people could put any questions you have in the chat, then either I can ask them to Shawn, or Shawn can read them, or you can say them yourself. But just to start off, I had a question about applications of some of the technology that you've talked about. Would it be possible, or is it possible to look at a single plant resolution? So, for example, there's lots of the people on board here will be geneticists, and we might want to differentiate between performance of individuals and populations, for example. Is that too high resolution to identify differences, or do you think you could do that? And you could look at populations and identify, yield potential in individuals, for example.
Well, I mean, I assume we would be imagining taking pictures of barley. Is that what you
Cereals.
think? I mean, when we talk about individuals, I also do forestry work. An individual tree is a nice large unit to look at. But we have very high resolution. I mean, I think we've flown 40 megapixel cameras on our drones, so we're getting centimeter scale resolution.
Yeah.
We can identify count heads of barley or ears of barley from a drone.
Yeah.
In an open field, and there's multiple heads per plant. If it's a continuous crop, it can be hard to separate one crop from another because they're all obviously all overlapping. But yeah, the leaf scale or the individual scale is definitely.
I mean, to be honest, I think the evaluation would probably start when there are small populations of either rows or F3 plots, maybe meter square plots or something like that. But it'd be kind of interesting to see if that would work. I mean, the other thing that we might think about is if you've got a field of 10,000 mutants, whether you can identify something which is, looking a bit odd, maybe photosynthetically or whatever.
Yeah, I mean, you could definitely, well, the idea of phenotyping is lots of tiny little plots, and then if it's a big field and you want to pick out something that's strange, sort of the precision agriculture perspective would be to look at the variability of that field and identify anomalies within it.
Sure. Thank you. Abdul Aziz, you have a question? Remember and unmute.
Yeah, sorry. Good afternoon, all.
Good afternoon.
Thank you, Shawn, for this interesting presentation. I have a question. In the use of the Fiji software for UAV image processing, does it require input for sorry, I'm trying to remember my question now because I did not write it up. Does it require input for atmospheric correction and geometric correction as well?
No, we're not applying that within Fiji. Now, in the case of taking pictures from the field, you're getting an image which is calibrated to the sensor. So, on our drones, we have calibration at least to reflectance at sensor. So, the impact of the atmosphere between the sensor and the drone could be 50 meters, 30 meters. So for these color indices with RGB or almost multispectral indices, it's not going to be a big deal. The only caveat I would put in there is if you have a very specialized sensor to estimate sun-induced fluorescence of chlorophyll using very narrow bands and radiance, then in that case, my colleagues who have done that have recommend atmospheric correction.
Okay, what of geographic correction? Geometric correction, sorry.
Geometric correction. So we usually use in a drone, we'll plan a flight and we'll take lots of pictures. And then the geometric correction comes from the algorithm that puts together the images and creates an orthomosaic. So that would be the geometric correction. Now,
so there will be need for a ground control point.
Yeah, or ground control points, or simply if you have a GPS on the drone, it will put together all of the images and it'll give you one mosaic that is ortho corrected. We call the orthomosaics orthometrically corrected. Sorry, my English is not doing great today.
So there is no need to worry about any geometric shift on the image at all.
No, we do try to take the images looking straight down at the crop. We have tested it with images taken sideways and diagonally, but whatever you do, you want to standardize your angle of capture.
Okay, thank you.
So there's a couple of questions in the chat. Francois, do you want to ask your question or shall I ask it for you?
I can ask it if that's. I just wanted from a beginner's perspective, and I'll just put on the video there enough to be. In terms of somebody wanting to get into this aerial-based phenotyping, what types of sensors would you recommend? I saw that you had a very high degree of success with the RGB. And would that be the main, or would you want to have some IR, infrared measurements on there? What would be your recommendation?
You know, I've been a big fan of the RGB, especially if you're just getting started. It's high resolution. The color calibration is quite good. The only thing I would add, and even if you're thinking that you're doing a study of heat stress or drought stress or something like that, in which color changes might affect it, you know, if you think about your plant physiology, the thing that goes first when there's water stress is that the plants grow less. So I really think that RGB at high resolution, a much cheaper sensor, is going to give you a really good estimate of fractional vegetation cover, good correlation with leaf area index. And if there's a disease or a rust or something like that, you're also going to be able to capture those small changes in color.
Great. Thanks very much.
And those are easier to process sensors, and they come at a tiny cost in comparison. Now, I've seen a couple of drones that have been combining thermal and RGB in the same sensor. And we have some of those, and we work with them. Now, we spend 5 times more money and effort trying to process the thermal images than we do the RGB. So it's interesting, it's useful, and it's a little bit more complex.
Great. Thank you.
No problem.
So it's lovely to see we have a couple of very curious Canadians on the line. Kelly, do you want to ask your question?
Curious Canadians.
Yeah. I don't know about. Can you see me?
We can't see you, but we can hear you.
Clear.
Okay.
We can hear you, Kelly.
Good. So I'm actually 62. I'm what you might call a foundational plant pathologist, but we're slowly getting into some of the remote sensing research. Mostly, I'm helping some colleagues imaging weed trials and looking at also crop safety in terms of different herbicides. But we run large-scale nurseries for the main foliar diseases of barley here in the prairie region of Canada, not for spot blotch, I might add. You know, we're looking at, we have DJ Mavic 2, I think it is, and then a red edge camera, but we're looking at imaging. And one of the concerns that we have is we run hill plot nurseries to try and minimize the amount of field space that we utilize, put in as much plant material, but it doesn't lend itself to providing enough space, I think, between individual plots. The other issue that they're a bit concerned about is imaging from above. And especially if we're looking at disease development over time. Typically, our ratings are done towards late milk, early doe, just because of time constraints, labor constraints, and so on. We run a lot of other trials. Imaging from above the upper canopy could potentially interfere with your imaging of the middle and lower canopy. It's really interesting to hear about the RGB potential. We have a multispectral camera on the drone. We're working with Kassav Singh out of Lethbridge with Ag Canada. That's who I'm based with in central Alberta at Lacombe. But we're wondering about imaging from the ground and the idea of looking at and we're looking at hill plots. And normally when we rate ourselves, we're looking at the whole circumference of the hill plot, bending the plants over, looking at disease symptomology, and we use a 0 to 9 scale to rate the severity of disease. But I guess two aspects of the question, sorry for blathering on for so long, is how far apart do we need to have our plots? And are there options in terms of imaging from the side? Ag Canada has purchased some of these phenocarts, but they're cameras that are mounted on a bar that look down from the top. So there might be similar issues that you'd have with the drone. Sorry, I'll quit there.
Okay, so let me see. So, you know, even when you're flying one of these Mavic drones, the cameras are usually 20 megapixels or something like that. So unless you're flying really high, you're going to get 2 to 5 centimeter resolutions of the image, or 1 centimeter resolutions of the image. We fly 30 or 50 meters, sort of depending on how we feel that day and if there's anything in the way and what the project's going to be. So I don't think the spacing is really going to be an issue. In terms of the size of the plot, sometimes in our phenotyping process and in our processing trains with the mosaic tool, and when we take pictures on the ground, we do try to eliminate at least the edge of the plot, because there's often an edge effect where you can see the wheat or the barley on the very edge is growing a little bit better than the ones in the middle, just because it's got a couple centimeters less of competition on the side outside of the plots. In terms of imaging from the ground versus imaging from above, from an aerial platform for canopy penetration, one thing to take in mind is that the further away you are from the subject when you take the picture, the less of an angle variability you have within the image. So if you're really far away, yeah, you have high resolution because you're using a good camera, but you're going to have everything pretty much from the same angle looking above. If you take the picture from really close, in that one picture, you have the whole plot, and actually you're getting oblique views on the side of the image, and you're getting a nadir view in the middle of the image, because the camera has got its own field of view. And on the side, maybe you're at a 20-degree angle, whereas when you look straight down, you've got basically a zero zenithal angle, right? So one of the things in classical remote sensing that was published maybe decades ago when I first started was that imaging at an oblique angle usually provides better canopy penetration. And what that means is that if you take the same resolution image on the ground, you're actually getting a combination of different angles of perspective in the image. So on the outside of your image, you're getting oblique angle views and greater canopy penetration. And besides resolution, I think that's really one of the points that is sometimes giving us better results from ground images. It's because it provides better canopy penetration and more information about the middle and lower canopy. On the other side, if it's not wheat, for example, I have a small side project in maize and corn right now, and these are taller plants. And using the 3D model, which we managed to produce at a resolution of about 5 centimeters for a maize UAV project, we were actually able to delete all of the weeds underneath the plants, underneath the maize crop, and then extract just the vegetation indexes from the maize itself from above, right? So you could similarly take the same idea and fuse three-dimensional imaging with RGB imaging and trying to get an estimate of the performance of vegetation at different levels of the canopy. If you want to do that, I would recommend getting a nice RTK GPS module and make sure your vertical precision is good.
Brilliant. Thanks, Shawn. Derek, last question.
Okay, I'll slip in two then. One's easy. I'm just curious to find out, is that a Jackson Electric you've got behind you? But that's the social one. The other one was actually, have you explored anything, Shawn, with using near IR sensors to advance towards crop quality assessment or compositional assessment in the system?
Yeah, so so that last study I showed with wheat phenoscale, we're actually using an 11-band multispectral camera covering I think like 4 or 5 bands in the visible and 4 or 5 bands in the near infrared. It also has a thermal camera, so we're catching thermal infrared, near infrared, and also visible and red edge images there. A lot of the near infrared plateau between sort of like 800 and 1,000 is just related to biomass. We use some of the radiative transfer model algorithms from leaf to canopy to look at that. We see that. There is a water content, canopy water content absorption at 950, 980, which we can catch. We have seen pretty good results of estimating protein when we have 11 spectral bands we can put those in there. And that's sort of a combination of carotenoid, chlorophyll, different pigments all together, along with leaf area index.
Okay.
Now, I did my PhD actually on airborne hyperspectral imaging in collaboration with NASA and the Catalan Institute of not space science. I forget. It was an acronym in Catalan. I have to remember. And we used hyperspectral measurements, and some of those were full range hyperspectral. And so I know there are some nitrogen specific, protein specific absorption features. I think maybe Lichtenthaler 92 did one, the nitrogen difference absorption index, or lignin. And you can get some of those other biophysical properties started showing up after around 1600 or so.
So do you ever take, just to follow that, do you ever take the response back and convert it into second derivative spectra so you can start to tease about even finer detail?
Well, you know, so the hyperspectral was my PhD. We just got financing to buy our first hyperspectral UAV through this multi-university consortium. So we'll start flying that next year. I've also tried to book some flights from hyperspectral full range flights from Avarice 4, which is now in Zurich.
Yeah.
And so if I'm lucky with that, maybe we'll be able to get back into hyperspectral. We did do some continuum removal and some first derivative work. Especially the first derivative was good for identifying the inflection point in the red edge, which is quite closely tracking chlorophyll, a little bit better than looking at broadband vegetation indexes.
Okay, thanks.
Pleasure.
Fantastic. Well, I see we're at 3 minutes to 2, so our hour is just about up. Shawn, I just want to thank you again for a great presentation. I think you've still got quite a lot of people online, so if you could just show your appreciation to Shawn, you'll thank him very much. Thanks so much, Shawn.
Pleasure. Pleasure. Oh, and that's a Geo Micro. It's a small sized electric guitar in the back. It's my daughter's. Anyways, thank you for having me. It's been a pleasure, and hope to see another seminar sometime in the future.
Yeah, great. Listen, thanks a lot. Bye, everybody.
Bye. So, take care.
Bye-bye.
Thanks.